Thank you, Di, and it's wonderful to be here. This is a fantastic meeting, and listening to the talks this morning and this afternoon, I've really learned a lot. And it reminds me that underneath all the wonderful clinical application and directions of travel that this discipline, that medical genetics and clinical genetics, this journey that it's gone on for these last decades, underneath that is a wealth of fundamental basic research. And as I've taken on this role as executive chair of BBSRC a couple of months ago, I'm very much reminded about that journey from fundamental research to its application and the importance of the community connecting and interacting and building those bridges. And with that in mind, actually, when I was asked to talk about epigenetics and epigenomics, it's quite a daunting task. So what I decided to do was to give a bit of a research story to talk about where I think epigenetics is sitting at the cusp of some really exciting conceptual advances in our understanding of phenotypic variation. We've heard a lot about genetic variation today, and I'm going to talk about the role of epigenetics. Towards the end of my talk, I want to talk about the role of epigenetics in this variable expressivity in outcome that we're hearing about. So this is actually some of the stuff I'm going to talk about is not published, and it is a sort of research talk, so I apologize if I'll try not to lose people along the way, but hopefully there'll be something for everybody. So as I say, epigenetics is a huge discipline. If you ask a dozen people what epigenetics means to them, you'll get a dozen different answers. So this is what epigenetics means to me. It's about the modifications to DNA and chromatin and their influence on genome function. They include things like DNA methylation, the addition of a methyl group to the fifth carbon in the C base within our genome, but also to post-translational modifications to the histone proteins, these core proteins around which the DNA is wrapped that compact our genome and lets it fit into our meters of DNA into the nucleus, into these bundles of chromosomes, but also these epigenetic modifications are extremely functionally relevant for how our genomes are controlled. And importantly, epigenetics happens in the nucleus. It's not about RNAs doing things in the cytoplasm. Epigenetics happens in the nucleus. It literally means on top of our genomes. So the three key things that epigenetic modifications evolved to do were to first contribute to the functional architecture of our chromosomes. Classic cytogeneticists, cytogenetics has shown us that our chromosomes are packaged into tight bits and light bits, and that actually can be manifest molecularly with an epigenetic description. Heterochromatin is tightly packaged with particular repressive histone modifications and DNA methylation, and regions of euchromatin, regions of open chromatin where genes need to be accessible to the transcriptional machinery of the cell, have a different set of epigenetic modifications that are associated with gene activity and openness. In addition, around our centromeres, if we don't package those regions around our centromeres properly with repressive epigenetic states such that you have a compact piece of architecture, you can't segregate your chromosomes properly during mitosis. So epigenetics has a very important role to play in the functional architecture of our chromosomes. It is dynamic. It contributes to the dynamic regulation, but also the heritable maintenance of gene expression within particular lineages. So you may have heard of the Waddington model, where you start off with an undifferentiated stem cell and you acquire a hierarchy of epigenetic changes that shape the gene behavior within cells and direct them down particular lineages. Very important in development. And then the third thing that epigenetic modifications evolved to do was to manage our repetitive elements. Now, medical geneticists don't think about the 40 to 50% of the human genome that is comprised of repetitive sequences. And I'm going to spend the latter part of my talk talking about that, because I think that they have an important role to play in phenotypic variation. So Unlike our DNA, our epigenome is dynamic from cell type to cell type, but it also has the ability to be stable. The marks, these epigenetic marks, are generated by enzymatic machinery that write the epigenetic state, that read the epigenetic state along with other proteins, other factors that respond to these chemical modifications that are sitting on our chromosomes. And also enzymes that can erase those epigenetic marks. And indeed, there are many genetic disorders that I chose not to give a talk on that today, that are associated with particular genetic diseases that influence the ability of our chromatin to be packaged properly. And indeed, many of these readers, writers, and erasers are perturbed in cancer. And there are some interesting therapeutics that are evolving that target some of these, this epigenetic machinery. I'm going to talk mostly today about DNA methylation. DNA methylation, this methyl group that sits on our Cs at the CpG dinucleotide within the mammalian genome, this is stably maintained during cell divisions. And that's because when that mark is placed on one strand of the DNA and the newly synthesized strand emerges, there's an enzyme that recognizes that, called a methyltransferase, and puts a new methyl mark opposite where that one was. So at the opposites, at the CpG on the other strand. And that means that you have an inherent heritability of a methylation state during the cell divisions, during the mitotic divisions within our somatic lineages. Now, that ability for an epigenetic state to be heritable has created a context in which it is believed that epigenetic marks can be heritable from one generation to the next. DNA methylation is mitotically heritable, but not in general inherited from one generation to the next if you're a mammal, because in mammals, most epigenetic states are erased not once, but twice from one generation to the next. And I'll show you a bit about that in a minute. So they're erased and then they're reconstructed between generations. So the ability to have an epigenetic memory from one generation to another, perhaps through an environmental insult, which has become kind of the dogma now, the environment talks to the genome via the epigenome and it can be transmitted from one generation to the next. Actually, that's a really tall order, given what I've just told you about epigenetic erasure and reconstruction. So I want to talk about two stories today. One is about genomic imprinting, which is a real paradigm of epigenetic inheritance. And the take-home message here is that in order to maintain a memory from an epigenetic memory from one generation to the next, it's actually quite difficult and it's highly regulated and highly evolved. The second story I want to talk to you about is about that repeat genome and how perhaps some of the phenotypic variation, the variable expressivity that we see in phenotypic outcomes in patients and actually in the normal population could be due to the epigenetic targeting of these particular elements. And I'm going to talk about that towards the end. Is it a paradigm? And actually we got into it because we thought it was a paradigm of mammalian epigenetic inheritance. And it turned out to not be a great paradigm of mammalian epigenetic inheritance, but rather a paradigm of variable expressivity. So genomic imprinting, this year we're celebrating the 40th anniversary of the discovery of this remarkable phenomenon, which is genomic imprinting, which is a process that was discovered independently by both Davor Salter on the left and Azim Sarani on the right in 1984. And I was fortunate to postdoc with Azim and indeed publish with Davor over the years. So what they did was they took a newly fertilized mouse egg, they removed the male pronucleus, that black circle there, and replaced it with a second female pronucleus to generate a diploid gynogenetic bimaternal mouse embryo. They could also do the same with the female pronucleus, and you can tell the difference between these pronuclei because they're different sizes. Plucked out the female one, and the one that came from the egg, and replaced it with a second male pronucleus that came from the sperm. And the resulting mice, when they were transferred into pseudo-pregnant recipient female mice, because you can do that in mice, ended up telling us that a biparental embryo could not make it to term. So on the right, you see the parthenogenetic or gynogenetic bimaternal. It makes a relatively normal embryo, but the yolk sac and extra embryonic tissues don't develop. And on the left, you see the bipaternal one, the one with two sets of chromosomes from dad. This is not about sex chromosomes. This is about autosomes here. Two from dad, and you get, actually very rarely do you get an embryo that even looks like that. Mostly you get a mass of trophoblast material, very reminiscent actually of the complete hydatidiform mole that many of you will be familiar with in human products of conception. So what this told us, this failure, it told us that actually being diploid is not enough. You need a set of chromosomes from mum and a set of chromosomes from dad in order to develop normally. And that the maternally inherited chromosome seems to be functioning differently to the paternally inherited ones. If you don't have a maternal one on the left there, a set of maternal chromosomes, you can't make a proper embryo. And if you don't have paternal chromosomes on the right, you can't make a proper placenta. But importantly also, it suggested that the maternally and paternally inherited chromosomes that came in from the egg and the sperm somehow know that they came from the egg and the sperm. They know that they should be functioning differently. And that's odd because if you're using mice, you're using genetically identical males and females. So it has to be something that's not genetic. And that was a very, very exciting finding because that marking of parental chromosomes to confer these functional differences was one of the first examples of an epigenetic phenomenon. And what I've shown you here is a context in which you have the germline inheritance of an epigenetic memory from parent to offspring. So over the years, we and others have been very excited about trying to use genomic imprinting to understand the epigenetic control of gene expression and repression. And you can see here we now know that this red chromosome and this blue chromosome are differentially marked such that a gene that is imprinted can be expressed from the maternally inherited chromosome and repressed from the paternally inherited one. And a gene can be, an adjacent gene, in fact, can be repressed from the maternally inherited chromosome and expressed from the paternally inherited chromosome. So you're functionally haploid. It's as if you only have one copy. Quite remarkable and epigenetically regulated. So this was, as I say, a useful paradigm for all kinds of stuff. Imprinting is amazing in its own right, but actually it's taught us a lot about how the epigenome regulates gene expression. So how does the transcriptional machinery of the cell tell the difference between these two chromosome homologs and activate one gene and not the other? So actually we know the mechanism quite well now, that in fact there's a series of what we call germline-derived differentially methylated regions. These are imprinting control regions. And they're established differentially in the male and the female germline, and then upon fertilization, they are transmitted such that the maternally inherited chromosome and the paternally inherited chromosome have different epigenetic marks on them. Now, how can that happen in a context where I told you that the genome is erased and reconstructed from one generation to the next? And that's what's special about imprints. So this is, I don't think this pointer is working. Do we have a pointer? Maybe I can use. Do we have a pointer?
Hold the top button down, it's a digital pointer.
Good, thank you. I wasn't pressing hard enough. So here you have the primordial germ cells, the cells that are going to give rise to the egg and sperm in a developing embryo. And as they migrate up the hindgut mesentery to find themselves in the developing gonad, they lose their DNA methylation. Actually, they lose most of their epigenetic marks. So their chromatin becomes, if you're a mouse, by about mid-gestation, a little after, you are epigenetically naked. And then in the male and the female germline, in the prospermatogonia and in the growing oocytes, you start to reacquire methylation actually to enable the egg and the sperm-specific genes to be expressed properly in those cell types. And then, upon fertilization in the zygote, lo and behold, the whole thing happens again. And you get a rapid loss of DNA methylation from the paternally inherited chromosomes and a slightly more protracted loss of DNA methylation from the maternally inherited chromosome. So this is an epigenetic erasure and reconstruction of most of the genome in the mammalian pre-implantation embryo. But what's special about genomic imprints is that if they did that, they would lose their memory of parental origin. So there has to be a mechanism that maintains that epigenetic memory. There are some repetitive sequences that also are heavily modified. They're always heavily modified. They never lose their modifications, and they're very, very refractory to any kind of change. But these imprints are special because they retain these methylation marks in an environment where everything is losing theirs. Now, if you do a methylation, if you do a methylome of sperm and a methylome of oocytes, in other words, a genome-wide map of all the methylation in sperm and a genome-wide map of all the methylation in eggs, you find actually that there's quite a lot of differentially methylated regions. The yellow stuff is unmethylated, the red stuff are methylated CPGs, and you can see that the oocytes in sperm have quite a lot that's heavily methylated, quite a lot that's unmethylated, and actually quite a lot that's differentially methylated, way more than there are imprints. So in actual fact, and what happens by the blastocyst stage is this starts to be resolved such that by around implantation, you have fully methylated around most of your genome. Actually, the default state within the genome is methylation. Everything gets methylated except where it can't. So actually what's really important for imprinting is that it's not the germline methylation that is the essential component of the epigenetic inheritance, but the retention of that differential methylation in the milieu of the newly fertilized egg. And I can't emphasize enough the importance of the milieu of the newly fertilized egg, and actually the unfertilized egg too, to create an environment in which the genomes of the egg and sperm are properly packaged later in development. So how does imprinting work then? What retains that memory in the milieu of the egg? Well, it turns out that there's two proteins. They're called crab zinc finger proteins, and I'm going to come to that in a minute because they're really important if you haven't heard about them before, and many people haven't because they're vertebrate specific. These crab zinc finger proteins target the methylated copy of an imprint coming in, and they recruit the methylation machinery to ensure that that methylation is propagated in a very targeted, highly regulated way at a time when the rest of the genome is losing its methylation. They're called ZFP57 and ZFP445, doesn't matter. But in the absence, when we create mouse mutants that have lost these two proteins, the imprint memory is lost. So epigenetic inheritance at imprints is a highly regulated process controlled by crab zinc finger proteins in the egg and early embryo. So what are these crab zinc finger proteins? You'll hear about them in the future if you haven't heard about them now. There's 350 of them in the mammalian, in the vertebrate genome. They're specific to vertebrates. There's loads of them. And they hang out in big clusters. And what they do is they target the repressive epigenetic machinery to particular regions of the genome, in this case, to methylated regions that are imprints in a sequence-specific and methylation-specific manner, but actually also they largely target the repetitive sequences that comprise half of our genomes. So they are recruiting DNA methylation and other repressive histone modifications in a sequence-specific manner to different parts of the genome. They're modulated by their zinc finger the zinc fingers that bind the DNA, and the sequence. There's 3 amino acids in these fingers, the fingerprint that is really important for that specificity. And they're very different between vertebrate species. In fact, between humans and mouse, of the 300 or so that there are, there's only 80 that are conserved. So they're very rapidly evolving, and they're very polymorphic between humans and mouse. So that, and as I said, one of the things that they evolved to do, and why there's so many of them, is that they're trying to target these transposable elements, these repetitive sequences that take up such a large portion of our genome. A couple of them have happened to be co-opted to regulate imprints, which don't look like transposons and repetitive sequences at all. And actually, many of them do target other unique portions of the genome. So this takes me to the next part, which is about transposable elements and variable DNA methylation, which we thought might be a paradigm of mammalian epigenetic inheritance, another model, but in actual fact, it turns out to be telling us something about the epigenetic control of variable expressivity. But first I want to say a little bit about transposable elements in the repeat genome. So this is a range of different species, and you can see that they all have different amounts of repetitive sequences. The red ones are these so-called retrotransposons, these sequences in the genome that have the ability to mobilize. Maize has 3/4 of the maize genome is made up of these repetitive sequences, and about half of the human and the mouse genome are made up of these sequences. They look like, many of them are called endogenous retroviruses because they look like viral sequences. And so, in fact, you could imagine this epigenetic targeting being kind of a host-parasite response to an element that has the potential to move around and invade the genome. So they are targeted, so this epigenetic regulation is very important. They're targeted post-fertilization by DNA methylation and this histone H3 K9 trimethylation. This is a post-translational modification on one of the histones that packages our chromosomes, and it's done via these targeting by these crab zinc finger proteins in vertebrates. The one I want to talk about at the moment, the class of transposable element, is called an intracisternal A particle. It looks like a virus. It has these long terminal repeats, these sequences at the end, and I'm not going to go into the sequence in any detail, but the key point is that this particular element, this LTR, contains genomic features that not only, promoters in particular, that not only have the possibility of regulating the genes within the element that enable it to mobilize, but also the adjacent genes that are close to where this element has jumped into the genome. So protecting the genome from these elements is really important by epigenetic modifications. And the reference mouse genome, I'm a mouse geneticist, and actually I wanted to say that when you're studying epigenetic variation, it's very difficult to do that in humans because humans are genetically so heterogeneous. But when you're working with inbred strains of mouse, you know that your epigenetic variation is really not genetically conferred. It's epigenetic. It's a true epigenetic variation. You're not complicated by noisy genetics. And many of these elements, there's 10,000 of this particular class in the C57 black 6, the black mouse genome, which is the reference genome, and many of them have lost the ability to mobilize. They're often they just hang around, actually as just solo LTRs. They've lost the rest of this stuff. So these solo LTRs are, even though they don't have the rest of the retro element next to them, are still targeted by the epigenetic machinery. So the model that I want to talk to you about is called agouti viable yellow. It's a classic mouse model, and this is the paradigm of transgenerational epigenetic inheritance and a couple of other things as well. So these 6 mice, are genetically identical, but you see quite dramatic phenotypic variation. There's a real something is making these mice look very different. And that something is the insertion of one of these retrotransposons upstream of the coat color gene agouti. And this gene, this LTR here, carries a promoter that drives abnormal expression of that coat color that makes the mouse yellow. And normally, most transposons are 100% methylated at that LTR. But this particular one has either no methylation or varying levels of methylation in between. So here is a yellow mouse that has virtually no methylation. It's driving lots of ectopic expression of this gene. And here's the brown mouse that is fully methylated and is expressing the normal coat color. And then there are these mottled mice that have different levels of methylation at this LTR. So a phenotypic variation that is not genetically conferred, but epigenetically conferred. This mouse is a poster child for all kinds of stuff. This allele is very sensitive to environmental factors. You can feed the pregnant female methyl donors, and you can shift the coat color of the mice to more agouti, to greater methylation and more agouti. You can feed the pregnant female bisphenol A, which is an endocrine disruptor, and you can shift the methylation, the coat color to yellow. And I mean, there's a bit of controversy in the literature about the environmental modulation, but it's generally felt that these alleles are sensitive to the environment. But more importantly, it was discovered by Emma Whitelaw and her team in Australia that a yellow mum is much more likely to give rise to yellow offspring than a pseudo-agouti mum, who is much more likely to give rise to brown offspring. And I've told you that the coat color is not genetically conferred, but epigenetically conferred. Therefore, this was a very exciting paradigm of epigenetic inheritance. So how does that happen when we know, I've told you, that the genome is erased and reconstructed from one generation to the next? Well, it turns out that the methylation at the agouti viable yellow allele is erased and reconstructed like everything else. So what is that mystery memory? We don't know the answer to that, and I'm not going to go into that in depth. But one of the things that we thought we might do was to ask the question, is this kind of behavior unusual? How many other repetitive elements in the mouse genome behave like this? Is this a common thing? And might that be able to tell us something not only about phenotypic variation and the role of epigenetics in it, but epigenetic inheritance? To cut a long story short, we found about 100 of these in the C57 Black 6 genome. Most are in the middle of nowhere. They're not adjacent to genes, but those that are influence expression in a methylation-dependent manner. They're relatively young mobile elements. These are more recently evolved elements. But we did a number of experiments to show that they were actually quite insensitive to environmental insult, unlike the agouti viable yellow. And in terms of their inheritance, this is a data slide. I don't want you to pay too much attention on it, but to say that we looked in sperm, each of these elements is 100% methylated in sperm, but the offspring of these males have the full repertoire of differential methylation. Each individual has a different methylation state. So variable methylation in the offspring, despite 100% methylation in sperm. So that says that, like agouti viable yellow, they're probably erased and reconstructed. But that actually doesn't, I mean, remember, this is not, the coat color thing doesn't say anything about mechanism. So we wanted to know whether these alleles actually could, these mothers and fathers could transmit a particular methylation state to their offspring. Turns out they can't in general. It's, this heritability is the exception rather than the rule. We found one of these alleles that has a significant maternal transmission. You can see the slope of the line there, where mother's methylation correlates actually by about 17% to the methylation level of the offspring. So we can quantify that heritability, but it's really the exception and not the rule. And it's telling us, I think, a little bit about what the mechanism might be. So what? So here are these black mice that have these variable epigenetic states, but they don't seem to do anything that important. We couldn't see any phenotypic variation associated with the behavior of these transposons at these genes. And they weren't showing heritability or environmental responses, but we were conscious of the fact that this is a laboratory mouse that's been bred in the lab for many, many years, and perhaps if we went to a more ancestral mouse, an ancestral laboratory mouse, we might learn a little bit more. And we chose to study to have a look in this mouse called Castaneus, which diverged about 500,000 years ago from the black mouse on the right here. There's 20 million SNPs that are different between black 6 and castaneous, and 8 million indels, very well characterized, reasonably well characterized genome. And we found a very different repertoire of transposable elements that were variably methylated within this older strain. I'm not going to go into this in a lot of detail, but they're a much broader repertoire of elements and a much broader class of repeats. So what is the mechanism that makes these retrotransposons not get fully methylated? It's just a small class of them. Most of the tens of thousands of repetitive sequences, millions in the mammalian genome, get properly targeted and properly shut off. What is it about these ones? And we noticed that when we crossed a black 6 mouse, this is where the genetics comes in, we cross a black 6 mouse to a castaneous mouse, some of these variably methylated transposons got 100% methylated, suggesting that there was something in the castaneous genome that modified the behavior of these black 6 alleles and caused them to behave like proper transposons. And so we back-crossed and we identified a single modifier locus that causes this hypermethylation. And we mapped the modifier locus to a cluster of crab zinc finger genes on mouse chromosome 4. So that suggested that not only were these crab zinc finger proteins different between species, but actually within species, you could have strain-specific differences, highly, highly variable family of proteins that you might expect from a protein family that were evolving to target a set of elements, repetitive elements that were constantly evolving in waves of infection of mammalian genomes. And that turned out to be the case. In fact, that we found that there were 2 particular clusters, one at the end of chromosome 2 and one at the end of chromosome 4, that had lots of these crab zinc finger proteins that were not at all conserved between different strains of mice. So these crab zinc finger proteins are highly variable. The interaction between this particular protein that's targeting the epigenetic machinery and the element that needs to be silenced is really important. And if you don't do this right, for example, if you have a new element, a new transposon that's shown up, or a crab zinc finger protein that's recently evolving, it doesn't quite manage to do the methylation properly. So is this important? We thought that it wasn't really in the black 6, and there's 2 questions really that this raises. First, is this important for outcome, for phenotypic traits, and what do humans do? So this is my only human slide, but there are hundreds of crab zinc finger proteins in humans as well. They hang out in clusters. The cluster on human chromosome 19 has 180 of these crab zinc finger genes, and we went into to the draft human pangenome reference that we heard a little bit about this morning, and profiled the crab zinc finger proteins of the 2,535 individuals from 5 different populations from that human pangenome reference. And what you can see, the first thing, I'm not showing you the data for this, but 60% of the crab zinc finger proteins in this cluster have a sequence variant in at least one of the individuals profiled. Highly, highly variable. But most interestingly, this is one of the crab zinc finger proteins, and this is it's profiled in the 5 populations. Each grey line is the amount of variation at each of these different sites. Some of them are not protein coding, but look at this last exon here, and in the African population. The African population, you see massive variability in this last exon, suggesting, in fact, that if you were to cross because I'm a mouse geneticist, if there was offspring born from these different populations, this particular crab zinc finger protein might not be able to target the transposons properly in the offspring of that hybrid mating. So that's, I think, pretty important. And I think that we need to be thinking about the possibility that this vast repetitive element, the vast amount of repetitive elements that are epigenetically targeted in our genomes by a highly variable family of targeting proteins could influence phenotypic variability. But to what extent do they influence phenotypic variability? So what we did was we quantified the expression of, in the Castaneous mouse, of the genes that were adjacent to these elements, but they didn't, and of course in BLAC6 they didn't have them. So we looked at differential expression between BLAC6 and Castaneous for the genes adjacent to these elements. And what we found was that there were 10 or so highly significant differentially expressed genes, 2 of which were very highly differentially expressed, that are very important for metabolism. So there's 2 things going on here. You've got a transposable element insertion adjacent to these 2 genes here. And you've got, and what that results in is differential expression between the 2 strains. So here you have normal expression in liver, ectopic expression in T cells. This is BLAC6, this is Castaneous. I don't need to tell you about reading transcriptomes, you don't need to know about all that. But basically you get differential expression that is associated with the presence of the transposable element of these 2 genes. What we see is that expression is inversely correlated with the amount of methylation at that transposon. I've got 2 slides left. I'm not going to talk about ADH2, just this ACAD12 gene, which promotes fatty acid oxidation. And just to tell you that Castaneous mice have significantly different metabolic outcomes compared to other inbred strains. Down here, you can see they have very low body weight. The green are the Castaneous. They have very low body weight compared to these other strains. And in actual fact, despite having a low body weight, their average food intake is very, very high. But what we noticed about this data set, which was published several years ago from the collaborative cross in mouse, is that there's quite a bit of variable expressivity of this behavior in these mice. And so we went into our mouse house and asked whether the methylation levels of these elements correlated with the body weight in the normal Castaneous mouse colony, and indeed it does. So in conclusion, what I've told you, I hope, is that epigenetic inheritance at genomic imprints is enabled by methylation-sensitive, a special pair of crab zinc finger proteins. It's highly regulated and it's targeted. That there is a repertoire of variably methylated transposable elements in different strains that is conferred by strain-specific members of this same family. But there is little evidence that that is epigenetic inheritance at these particular loci. So it's the methylation that's driving it at imprints. These clusters are divergent between different strains of mice and, importantly, between different human individuals and different human populations. And the inter-individual differences in this methylation can confer variable expressivity of phenotypes in genetically identical mice. And we can postulate, perhaps, that epigenetics can potentially underlie the variable expressivity of normal and disease phenotypes in humans. And I'll stop there, and thank you for your attention. The work that I presented has been done predominantly by Kevin Costello, and thank you.